So I want to thank the meeting organizer for the invitation. I'm here to present the strategy we use for proteomic characterization. So why proteomics and protein post-translational modification? Let's show in this figure that in the cells, normal cell and the cancer cell perform their normal functions. Oh, sorry, I'm going the other way. By gene expression, the protein level, as well as protein formed modified by protein post-translational modifications. Cancer proteomics, protein genomics is being used to characterize cancer in a comprehensive level and link genomic alterations for the functional mechanism of cancer pathology and delineate the novel insight that are only captured by proteogenomic characterization and generate data to allow investigators from biology and the clinical side to further investigate their questions. As one of the protein characterization center, like Henry talked about, Johns Hopkins University is being characterized cancer tissues using proteomics approaches by global proteomics and phosphoproteomics, glycoproteomics, and cytolomics and ubiquinomics. So using this approach, we've been characterized 4 of the cancer type Henry described in his presentation. How did we do this? So this slide shows that the protein from the tumor samples are digested to a protein fragment we call peptide, and the peptide carry modifications including glycosylations or gluconide modification or phosphorylation example, or as well as the peptide without any modifications are generated for characterization by mass spectrometers, which measure the peptide mass and their modification by different chemical modification group. So the modified peptide can be further enriched using chemical, enzymatic affinity, or chemical physiological approaches to further enrich the comprehensive characterization of protein post-translational modifications. The peptide with or without modification further separated by liquid chromatography, and the peptide separated, eluted from the HPLC can be measured by mass spectrometer by their precursor mass, and a certain peptide with specific mass are isolated by mass spectrometer sent to the collision cells to generate fragment ions, further measured by mass spectrometry to determine the fragment ions, as well as the ions generated by modifications. So this spectrum we call MSMS spectrum can be used to correlate with a database where the sample are generated from, or correlated to the spectral library generated from the spectrum using the software tools, as we describe here in the software development publication. So although cancer tissues has been characterized by genomic transcriptomics by TCGA and a number of other studies for tens of thousands or like hundreds of thousands, and tumor protein genomic characterization of cancer is just the beginning. So far, there are 11 cancer types, roughly 1,000, 2,000 cases have been characterized by protein genomics. Johns Hopkins Protein Characterization Center has been involved in ovarian cancer, kidney cancer, head neck cancer, and pancreatic cancer characterizations. So in the next few slides, I want to show that how our protein genomic characterization can be used to characterize a retrospective sample from TCGA collection and characterization, as well as the prospective collection by CPTech. So this is our initial involvement, our CBTAC, which is the characterization of ovarian tumor, was characterized and collected and characterized by genomic and transcription characterizations by TCGA, which was published in 2011 in Nature. Our characterization, together with Pacific Northwest National Laboratory, characterized the proteins as well as glycoproteins, phosphoproteins, cetylated proteins, and determine the PARP inhibitor sensitive cells, cancers related to histone acetylation. So in the next few slides, I want to summarize our recent study, which still use the same material collected and generated for our original proteomics and the phosphoproteomics study for glycoproteomics studies. So in our glycoproteomics study, we find that the glycoproteomics data can separate the ovarian tumor to 3 subtypes. The 3 subtypes is correlated with the highly overexpressed cetylated glycoproteins in subtype 1 and overexpressed oligomannose modified glycoproteins in subtype 2 and fucosylated glycoprotein in subtype 3. When we overlay the glycoproteomic subtype with the previous proteomics analysis and the TCGA study, we can see that the subtype 1, which is a highly cetylated subtype, is correlated with MRA and the protein subtype of differentiated tumors. The subtype 2, which is highly overexpressed in the oligomannose, is correlated with immune reactive and proliferative subtype. And subtype 3 is related to mesenchymal subtype. So this overexpression of the glycopeptide can be further investigated to determine the genes that involve in this overexpression. Furthermore, there are over like hundreds of glycosylation genes responsible for glycosylation biosynthesis. The correlation study showing that the oligomannose glycoproteins are correlated positively with PRK SH1 and negative correlated with MAN1A1, and fucosylated glycoproteins are correlated, positive correlated fucosylated transferase and negative correlated fucosidase, and cetylated glycoproteins are positive correlated with one of the cetylated transferase, ST3 GAL1. So this has been observed in both proteomic level and RNA level. To summarize what we find from the glycoproteomics study, we thought this is happening in the ovarian cancer 3 subtype. In one of the subtypes, somehow this distorted, but in one of the subtype with oligomannose overexpression, we saw this synthesis of oligomannose structures by removing one of the glucose from the unglycan precursor to MAN9, and the glycosylation enzyme MAN1B1 responsible for overexpression of MAN1, the oligomannose overexpressed glycoproteins, and fucosylated peptide is correlated with FUD11 and fucosidase 1, and the cetylated peptide is correlated with ST3 GAL1 genes. So this is summary what we observed from the glycoproteomic study of ovarian cancer. So recently we characterized the CBTAC prospectively collected pancreatic ductal adenocarcinoma among 140 pancreatic tumor, 67 normal adjacent tissue, and 9 normal duct tissue characterized. This is represent the simultaneous generation of genomic, transcriptomic, proteomic, phosphoproteomic, glycoproteomics data. Instead of the ovarian cancer study, just show that the sample collection happened over 10 years ago, and genomic also published over 10 years ago, but the proteomics and the glycoproteomics happened in a recent study. The pancreatic cancer study actually allow us to simultaneously investigate all the data generated in this same tissue. So this study actually used an orthogonal approach to determine the cellularity of the cancer cells using RNA-seq data, methylation data, as well as the data deconvolution studies. And protein genomic characterization reveal the functional impact of genomic alterations and the multi-omic subtypes. Protein genomic study uncovered putative therapeutic target for kinase and downstream of KRAS, which is difficult to drug for the target. And a multi-omic study also reviewed the endothelial remodeling and the glycolysis to immune cell exclusion, and identify inventory of proteins and glycoproteins responsible for cancer can be used as a diagnostic or therapeutic intervention. So the most importantly, this study also review the data or make the data available, including to public, including the raw data and bioinformation friendly downloadable data, as well as data analysis tools developed or utilized in this study into the public. I want to end my talk by acknowledging people who involved in this study, including people from Hopkins, clinicians, and data analysis people, and data generation, and the CPTAC consortium. Especially, I want to acknowledge the Baylor and Washi group, called PGDAC group, for the pancreatic cancer studies, and the entire CPTAC consortium and program director like Henry and Maddie, Tara, Emily, and Chris, as well as other PCC and the CPTAC PGDACs for this collaborative study. I want to acknowledge the support of NCI CPTAC for this data or result I present here. Thank you for your attention.